Right. This is Dr. Ben Wagoner down here in the basement of Lewis Science Center. And let's look at the second part of what I have to say about viruses. Now that we've covered transcription and translation, you're in position to understand much better what viruses do and how they can be defended against, both by your own body's defenses and by drugs that have been developed to combat virus activity in the human body. Hopefully, this will be of interest and timely and maybe a little reassuring at this time. You defend against viruses using a population of white blood cells. Generally, these are called lymphocytes. More specifically, this is a population of cells called cytotoxic T cells. They move freely through the body, and they can even slip in between cells in many of your tissues. They can wander wherever they need to go, and they detect virus-infected cells and bind to them and cause them to break up. They destroy virus-infected cells before the infected cells can release more viruses. Incidentally, they also defend against cancer. This picture shows in orange a cytotoxic T cell snuggling up to a cancer shell. Come on, Wagoner. Cancer cell shown in magenta. And the cancer cell is breaking apart into little blobs. This is what cytotoxic T cells do. They would do the same thing to a cell that was infected with viruses. Antibiotics block bacteria. They block bacterial metabolism, like bacteria making cell walls, things like that. Antibiotics do not work against viruses because viruses don't have a metabolism to block. Viruses don't do anything on their own, so there's nothing for antibiotics to work on. So taking antibiotics when you've got a virus infection does nothing. Maybe it makes you feel psychologically better, but it doesn't stop the virus at all. There are some drugs that work on viruses. One of them is called interferon, and this is what it looks like. It's got lots of squiggly parts. Those are the alpha helices of its secondary structure. And we don't need to go into detail as to how it works, except it stimulates the immune system to step up its game against a virus infection. So that's sometimes used for treating very serious ones. And there's some antiviral drugs that are worth looking at because we've just covered nucleotides. Up there at the top, you can see a real nucleotide. It happens to be guanidine phosphate. You can see the phosphate group is in tan, and the deoxyribose sugar is in light green, and then in brighter green you have guanine, the base, the one we abbreviate G. If you look at the bottom, you can see a compound called acyclovir, and it's got a guanine base, and it's got a fragment of that sugar, but it doesn't have the whole thing. If a cell tries to use this in DNA replication or in transcription, the process will be blocked. It would be kind of like sabotaging an industrial machine by feeding it the wrong parts to use. A cell that is filled with acyclovir is filled with fake nucleotides, and if it tries to use those, it'll shut down. It can't incorporate those into strands of DNA or RNA and make them any longer because there's nothing for the next nucleotide to attach to. So acyclovir is commonly used to treat the herpes virus, both genital virus, but I guess you could also use it for oral herpes, cold sores. This is another one right here. Has the fancy pants name of dideoxyinosidine. The trade name is Videx. And you can see, first of all, it's got an odd base. You haven't seen this base, the thing that's labeled in purple. The base is called inositol, and don't worry about that. More importantly, the sugar part of it does not have any oxygen sticking off it. It's not ribose, it's not deoxyribose, it's dideoxyribose, meaning that it has nothing it can use to link to other nucleotides. So if you feed this to a cell that is making lots of DNA or RNA, you block DNA synthesis or you block RNA transcription. That can have some side effects on the cell, but you can block virus replication this way. If the cell is cranking out new virus DNA or virus RNA, you can sabotage the process by feeding a cell lots of Videx. And Videx is one of the cocktail of drugs that's used to treat HIV. Those are all the pills that one HIV patient has to take each day. They don't treat HIV with one single drug. They treat it with a cocktail. Some of the cocktails are fake nucleotides like Videx that block transcription or DNA replication. There's other drugs that block the fusion proteins that we talked about and stop the virus from attaching to the cell. There's still others that can block the virus from assembling inside the cell, and even others that block newly made viruses from breaking out of the host cell. So the best way to treat HIV is not one single drug, but several, each one of which targets a different thing that the virus can do. You could use all of these strategies to treat other viruses. For example, severe flu can be treated with a drug called oseltamivir, trade name Tamiflu. Tamiflu works by blocking flu viruses from breaking out of the host cell. How this plays into coronavirus treatment, I don't know. It's still early days as we try to come up with vaccines and better treatments for it. I'm not sure whether antiviral drugs like this are going to be in use, but it's certainly possible. Now, I'm going to go ahead and stop there, and we'll do another lecture to continue because I don't want to overwhelm anybody's download capabilities. Talk to me at benw@uca.edu if there's anything I should be doing or if you have any questions. We'll get through this together. Stay safe.